Can somebody confirm you can hear me? I have a speaker error once again.
I can hear you.
Thank you. Okay, are there any questions before we begin?
I have a question.
Uh-huh.
It's regarding the unknown project that we have. Um, will you know the unknown list that we all have the document? Is the unknown bacteria one of those off the list?
If you're referring to
Or is it like a process of elimination?
it's one of the species on
The
the
list that
list.
has like 24 on there?
No, there should be like 40. There's something like 20 gram positive and 20 gram negative, something like that.
24, correct?
You mean
I
24
see
gram positive
24,
and
the
gram
list that's
negative?
attached. For a total, yes. The one that says 2019.
Can we discuss this during the lab?
You're breaking out. Can you say that again?
Can we discuss this during the lab?
Yes.
Obviously, I'm not sure what list you're referring to. All right.
Okay.
All right.
Mhm.
Are there any other questions concerning the lecture? All right. If not, let's get started. Today is April 13th. Excuse me, not April 13th, August 13th. And we should be starting chapter 8. I'm, I'm not sure if we started chapter. Yes, we did, didn't we? Let me get to chapter 8. I did get a speaker error, so if you're having trouble hearing me, just have me ask me to repeat something. All right, let's share my screen here. Come on. Oh, there it is. It was a long time before it gave me the option to share. I don't know why the computer would be running slow. I've got everything off except for Zoom. And obviously this one program where I have that open. But today is August 13th, and we should be starting chapter 8. Let me warn you, you do have a quiz 4 this week. Quiz 4 will be covering lab 7 and chapters 5 and 6. Any question about any of that? We do have a lab today. I'll discuss lab 9 and the lab time. All right, let's go to here. So in chapter 8, I've got that we had discussed DNA replication to here, that the DNA is made in the 5 to 3 prime direction, made on two strands, the leading strand, which is synthesized continuously. The lagging strand is synthesized discontinuously, resulting in many Okazaki fragments. And this is done by RNA polymerase. Helicase separates the two strands of DNA, and then DNA ligase joins the, I don't know, the disjointed pieces of DNA that have been synthesized together. All right, so let's talk about RNA transcription. Transcription is the synthesis of RNA from DNA. DNA is transcribed to make RNA, and you can make messenger RNA, tRNA, or rRNA, and those are also known as mRNA or messenger RNA, transfer RNA or tRNA, or ribosomal RNA or rRNA. Here's a question for you. when the RNA polymerase is transcribing RNA from the gene, how does the RNA polymerase know which RNA to make? Messenger RNA, tRNA, or rRNA? Nobody knows? This is something you should know from genetics. And that is the rRNA polymerase doesn't need to know because it depends on which gene it is transcribing. If the gene codes for protein, it'll transcribe it as messenger RNA. But if the gene codes for tRNA or rRNA, it'll transcribe it as tRNA or rRNA. Here's another question for you. What's the difference between messenger RNA, tRNA, and rRNA? Well, is there any difference in the nucleotide bases, meaning like A or U or C or G? That's a yes or no, people. All right. Is there any difference in the ribose sugar? That's a yes or no, people.
Yes.
There's a difference in the ribose sugar. So why is it called
No.
ribonucleic acid if there's a difference in the ribose sugar? There is no difference in the
No.
ribose sugar. All of them have ribose. All of them have the same nucleotides, messenger RNA, tRNA, and rRNA. There's only one difference, and that is the sequence of nucleotides. Depending on the sequence of nucleotides, it'll be messenger RNA or tRNA or rRNA. Most of the genes code for protein, so they will be transcribed as messenger RNA. But there are a few genes that codes for tRNA, and they'll be transcribed as tRNA. And there are a few genes that code for rRNA, and they'll be transcribed as rRNA. And this should tell you something too, and that is transcription does not happen to all of the DNA. Only the genes are transcribed. When we're talking about DNA replication, all of the DNA is replicated. But when we're talking about transcription, only the genes are transcribed. Remember, in eukaryote cells, most of the DNA is not or are not genes. Most of the DNA does not code for a gene in a eukaryote cell. In bacteria, most of the DNA does code for a gene. But in eukaryotes, most of the gene is actually non-coding DNA. Any question about any of that? Okay, transcription begins when RNA polymerase binds to the promoter. The promoter is a region of the DNA upstream of the gene that's going to be transcribed. And what the promoter does is it simply is a sequence of nucleotide that codes for the RNA polymerase to bind to it. Any question about any of that? And then the RNA polymerase moves down the DNA, transcribing RNA in the direction 5' to 3'. So RNA is made from 5' to 3'. And it moves down the DNA until the RNA polymerase reaches the terminator. Transcription stops when RNA polymerase reaches the terminator. What the terminator actually codes for is for the RNA polymerase to fall off the DNA. And once the RNA polymerase falls off the DNA, transcription has to stop. And then the RNA is released from the polymerase, and the RNA polymerase can then bind to another promoter and repeat the process. Any question about any of that? In the eukaryote cell, the RNA will need to move through the nuclear membrane to get into the cytoplasm. And once in the cytoplasm, protein translation can then begin. All right, this is actually not an electron microscopic image, but a, oh, what do they call that? A photo, it is an electron microscope which is taking the picture, but it's not using electron microscope directly. It's using antibody, I forget what they call that, but making the RNA polymerase light up photo something electron microscope, and then the DNA is actually lighting up. That's the DNA there, and right there you can see a little bit of the RNA being transcribed right there. Any questions about RNA transcription? All right, let's go into it in more detail. That slide's showing you. I think I've got a little video. Let's see if this works. Come on. Up. I have a feeling it's the wrong video. Yep, that's the wrong video. I think that's the one I want.
As I'm in the midst of applying for residency right now, wish me luck.
You have to
Grammarly has
have a
again
slight
been a
commercial.
lifesaver with my application.
There we go.
Egg proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and
Okay, this only happens in eukaryotes, by the way, having introns and exons, and have to be removed in eukaryotes.
modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation.
All right, we're not going to go into translation, so let me start by here. They mention that the DNA becomes a single strand during transcription. And they didn't mention, but it's a helicase that actually opens up the DNA. So both in DNA replication and in transcription, a helicase opens up the two strands of DNA, making a single strand for RNA transcription to occur. And then right here, they do show it right there. When the RNA is first made, it binds to DNA. But the binding, it doesn't happen for very long. And then the RNA flops off the DNA. And then let's see if that shows.
Polymerase links nucleotides to the
right there.
3' end of the growing.
So the RNA will initially bind to the DNA, and that's just going to be hydrogen bonding, the same as DNA bonding to DNA, double strand DNA. And then that falls off, and that allows the DNA to hydrogen bond to the other strand of DNA. And so this single strand of DNA is not that long in transcription because it snaps back to double-stranded DNA about right here. Any question about any of that? All right, so transcription begins when the RNA polymerase binds to the promoter, and then the RNA polymerase moves down the DNA, and it's the helicase right here, which makes the DNA single-stranded. So only one strand is transcribed, at least in eukaryotes. And that'll be the strand that contains the gene. And the RNA initially will hydrogen bond to the DNA, but it falls off. And after it falls off, the DNA can become double-stranded as the RNA polymerase moves down there. This DNA will hydrogen bond to itself and become double-stranded again. And that happens until the RNA polymerase reaches the terminator. Once at the terminator, where is that shown here? The RNA polymerase falls off the DNA. The RNA is released, and then the DNA becomes double-stranded again. Any questions about RNA transcription? All right, so the RNA will then move into the cytoplasm in a eukaryote cell, has to move through the nuclear pore. In the prokaryote cell, the transcription is happening in the cytoplasm because there is no nuclear plasm. And so the RNA is already in the cytoplasm and it can be translated. Translation is the synthesis of protein by ribosomes using the information in messenger RNA. Messenger RNA is translated in codons, that is a group of 3 nucleotides. And how that messenger RNA is actually read is it's read by tRNA. We say that the ribosome reads it, but actually the ribosome just moves down the messenger RNA, and then it allows the tRNA to come in and bind to the messenger RNA. The tRNA has an anticodon, which is 3 nucleotides, and the anticodon is the complementary codons to the codon. So the anticodon of the tRNA can hydrogen bind to the codon of the messenger RNA. And how that happens is the ribosome holds it all together, makes a little pore for the tRNA to come in, and then bind to the messenger RNA. So translation of messenger RNA begins at the start codon, and the start codon is the nucleotides AUG. So when we first The ribosome first reads the messenger RNA. Any part of the messenger RNA upstream of the AUG start codon will not be translated. Any question about any of that? So there's a messenger RNA. We don't have AUG being shown, but let's make it in the middle of the messenger RNA. The first part of the messenger RNA will not be translated. Translation will not begin until the ribosome moves to the start codon AUG. AUG codes for something else besides start protein translation here. AUG also codes for the amino acid methionine. So all proteins will always have their first amino acid methionine. Any question about that? When it's translated, all proteins will begin with the amino acid methionine. What happens if you have a protein that they don't want it to start with methionine? What do you think happens? You've got several proteins in your cells which do not want to start with the first amino acid being methionine. What does your cells do? I should look that up, but I'm afraid I don't know any protein off the top of my head which do not start with methionine. When the protein is first translated, it will have methionine in position number 1. But if the protein doesn't want that, it will simply remove the methionine. And we'll talk about modifying proteins, or have we already talked about that? How that happens is the Golgi apparatus modifies the proteins. And that's one thing the Golgi apparatus does. It will remove methionine if the protein doesn't want methionine on the protein. It can do other things like methylate the protein, add a poly-A tail, things like that. Actually, that's not the protein, that's the messenger RNA. All right. So once the ribosome reaches AUG, it begins protein translation, reading the nucleotides in as a group of 3, a codon, to translate each codon into an amino acid. And that process will continue until the ribosome reaches one of the stop codons. There are 3 stop codons, and you don't need to memorize them. You should memorize AUG because there's only one, but the 3 stop codons are UAA, UAG, and UGA. And like I said, you don't need to memorize those. Once the ribosome reaches a stop codon, the ribosome will fall apart, protein translation will end, and the protein is released from the messenger RNA and the ribosome. Any question about any of that? All right, let's look at the codons. Let me blow this up so you can see it. Each messenger RNA codon codes for 1 amino acid. However, organisms typically contain 20 different amino acids, like humans have 20 different amino acids. But yet, 64 codons are possible when you take all of the 3 nucleotide bases and stick them all together. There's 64 possible codons. This means that there's more than 1 codon for an amino acid, like phenylalanine has 2 codons coding for that same amino acid, phenylalanine. UUU and UUC code for that amino acid. Serine, on the other hand, has 4 codons coding for that one amino acid. Leucine has 6 codons coding for that amino acid. There's only 2 codons that only have 1 codon coding for the amino acid. And that is AUG coding for methionine right there. And then you don't need to know this other one, but it's tryptophan. That amino acid is only coded by 1 codon. Any question about any of that? So that means that the genetic code is redundant for which amino acid they code for. And that is known as the degeneracy of the code, that more than 1 codon can code for an amino acid. And if you look up the word degeneracy, that's one of its meaning. It's not meaning that the codons are a, what do you call it, a degenerate individual where they are undecent and immoral, degenerate that way. It means simply that there's more than 1 codon coding for it. Any question about any of that? How are you guys hearing me?
Yes.
Okay. All right. So that is translation. Let's watch a little video on translation. Now, I think this will be the one that we were trying to get, that tried to play earlier.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, The ribosome dissociates, and the newly formed protein is released.
Okay, any questions about that? If not, let's talk about protein translation in a little more detail. Protein translation requires 3 RNA types. There's the messenger RNA, which codes for the amino acids in a codon. There's the tRNA, which brings in the correct amino acid, and it has an anticodon, which actually will bind to the codon, and it's simply a hydrogen bonding. And it's the ribosomal RNA, which has ribosomal RNA and protein in the ribosome, which binds to the messenger RNA and then moves down it, allowing the tRNA to come in. And the ribosome has 2 or 3 pores in it. The third pore really isn't used, but the first 2, the tRNA will come in and be held by the pore of the ribosome and then come down and hydrogen bond to the messenger RNA. And that's what the ribosome does. It holds everything together and allows the tRNA to come in, stabilizes the tRNA for the tRNA to bind to the codon. Any question about any of that? All right. So upstream of the AUG, the ribosome doesn't do anything, meaning upstream of the AUG codon, that region of the messenger RNA is not translated. Translation will not begin until the ribosome reaches the codon AUG. At AUG, it tells the ribosome begin translation here, and then the first tRNA will come in, the first pore, which is shown in the dotted line here, come through the ribosome and then hydrogen bond to the codon of the messenger RNA, and it's the anticodon which binds to the codon. The first codon always codes for an amino acid, methionine, meaning the first tRNA will bring in the amino acid methionine, and that amino acid is just linked to the, let's call it the tail end of the tRNA. The second pore of the ribosome will allow the second tRNA to come in, and it will hydrogen bond to the second codon, and it has the correct anticodon to do that. Any question about that? So the second amino acid with the tRNA approaches and will bind to the codon. Then the first amino acid on the first tRNA will be removed from the tRNA, and that amino acid will be covalently linked to the amino acid on the second tRNA. And that's a peptide bond that forms between that amino acid and this amino acid. And we did mention that this is the only RNA enzyme that we will be discussing, that the enzyme that removes this amino acid here and then links it to that amino acid is actually a RNA enzyme that's part of the ribosome. So it's part of the RNA in the ribosome. Any question about any of that? And that's the only RNA enzyme that we will be discussing. at least in this class. Once the amino acid is removed from the first tRNA, that first tRNA can then fall out of the ribosome and leave the ribosome. And when it leaves, that will signal the messenger RNA to move down a codon. And when that happens, the third tRNA will come in. And an interesting thing that happens is what used to be in the second pore of the ribosome moves to the first pore, and let's just say the tRNA moves to the first pore. It doesn't come out of the second pore. It's just that I don't know how it happens, but the tRNA moved from the second pore to the first pore when the ribosome moves. And so these two pores must have a channel between them because the tRNA is still binding, hydrogen bonding to the codon of the messenger RNA. Any question about any of that? Anyways, that will then free up the second pore for the second tRNA to come in and bind to the third tRNA to come in and bind to the third codon, bringing in the correct amino acid. Once again, the rRNA enzyme will remove the amino acid from the tRNA and then link it to the newly added amino acid. And that's going to be a peptide bond, meaning a covalent chemical bond. And when that happens, this tRNA can then fall out of the ribosome. That will again signal the ribosome to move down one codon. And then what used to be in the second pore will move to the first pore. And that'll allow the next tRNA to come in the empty second pore of the ribosome. And so the ribosome will move down the messenger RNA, reading each codon, allowing the correct tRNA to come in and bring in the correct amino acid. You can notice that we can find AUG as a codon lower down in the messenger RNA, in which case the AUG codes for the amino acid methionine. It may or it may not code for the protein translation to begin here. And I don't want to talk about that, but sometimes in a protein, the ribosome, for some reason, doesn't begin protein translation here and will begin it there. And that's just some proteins want to have two types of the protein, one a bit longer than the other. And so that protein will be actually beginning protein translation here, as well as on another messenger RNA, it can begin it there. So you get two proteins of different sizes. And we're not going to talk about that, so you don't need to know it. But like I said, the AUG later down from the first AUG codes for the production of the amino acid methionine. Any question about any of that? All right, so the ribosome then moves down the messenger RNA, moving what the tRNA was in the second pore, moving it to the first pore, and this tRNA falls out of the first pore, which actually is a signal for the ribosome to move down one codon. And that will happen until the ribosome reaches a stop codon. Once the stop codon is reached, protein translation ends. How that happens is the stop codon actually codes for the ribosome to fall apart. That will release the protein from the ribosome and the messenger RNA. And obviously it ends protein translation. Any question about any of that? All right. So let's look at protein translation in an electron microscopic picture. Here you can see, this is in a prokaryote, you can see there is a messenger RNA, and all of those clumps on it are actually ribosomes. And so this is the artist's rendition of it. And as you can see, there can be more than one ribosome on the same messenger RNA. And that's just a bit of a complication. You can have more than one ribosome translating the same messenger RNA. At the same time, obviously let's see, which one would be the one that would have the longer? Is it that one or this one? I can do it from another way. That is here, so yeah, actually this one. This one here has been on the longest, so it'll have a longer polypeptide chain than that one. This will be the shortest polypeptide chain. And they're not showing the protein here. I'm not sure why that's the case, but they're not putting it in. So this one will have the longest protein. But the point is, even in a eukaryote, you can have more than one ribosome binding to the same messenger RNA. And that's just a complication that protein translation has. Any question about that? In prokaryotes, you have another complication, and that is the messenger RNA is being transcribed in the cytoplasm because that's all the prokaryote has. They don't have a nucleoplasm. And as you can see here, here's a long messenger RNA. There's a shorter messenger RNA, even shorter, shorter, and very short messenger RNA. And they're trying to show it here. Shorter messenger RNA. The messenger RNA would be, let me see if I can blow that up. I think it's the blue line. Yeah, the messenger RNA is the blue line running running down here. So that's a shorter messenger RNA than this one here. Oops, too far. The point is that in prokaryotes, before the messenger RNA is fully transcribed, you can have protein translation beginning. And that's what's being shown here, because that's the long messenger RNA. They're getting shorter. And right there is the beginning of the gene. So that's the shortest messenger RNA. The gene probably ends someplace after here. And that's the longest messenger RNA. The point is that protein translation can begin in a prokaryote cell, such as a bacteria, before RNA transcription ends. And this messenger RNA is still being made. And only down here does it become this long. So right there, it's a real short messenger RNA. Any question about that? You don't have that complication happening in a eukaryote because in a eukaryote, the messenger RNA is made in the nucleus, and only when the messenger RNA is finished being transcribed will it leave the nucleus and enter the cytoplasm. But once it's entered the cytoplasm, you can have more than one ribosome on that one RNA. And then all of these ribosomes are translating that one messenger RNA. Any question about any of that? So in prokaryotes, we have a complication, and that is before RNA transcription ends, protein translation can begin. In eukaryotes, we have another complication, and that is when the RNA transcript is first made, it will include regions of the RNA which code for the protein and regions of the RNA that do not code for the protein. This region is called an exon. This region is called an intron. The introns have to be removed before that messenger RNA will be allowed out of the pore of the nuclear membrane. So there's a check right here. And if that messenger RNA has an intron in it, that messenger RNA will not be allowed to leave the nucleus. What happens is that this original messenger RNA transcript has to have the intron spliced out and the exons spliced together. Splicing just means cut and then join. And they showed you in the video how that happens. And then this final messenger RNA, which has the introns removed, can then leave the nucleus. And that's just a complication in eukaryotes. Any question about any of that? We're not really going to talk much about introns, but in a eukaryote, within the gene, we have regions of the gene that do not code for the final product, meaning the protein if this is messenger RNA. And that's called an intron. In bacteria, the genes do not have an intron. But in eukaryotes, they do. Any question about any of that? And the other complication is eukaryotes have most of their DNA not coding for a gene. And then even in the gene, some regions of the gene do not code for the final gene product, meaning if it's a protein, it doesn't code for the protein. And that's just the way eukaryotes are. Any question about any of that? All right, if not, we've talked about the structure and function of genetic material. We talked about the flow of genetic information. We talked about DNA replication, RNA transcription, and protein translation. We're now going to talk about the regulation of bacterial gene expression. We'll use the operon model to talk about gene expression. And then we'll talk about mutation, genetic transfer and recombination, and we'll end talking about, it's only one slide, genes and evolution. So in a bacterial cell, the cell has ways to regulate its cell contents. One way for the cell to regulate its contents, meaning what the cell's making and what inside the cell, one way to regulate the cell's contents is to control gene expression. If a gene is not wanted to be turned on, the cell can turn that gene off, and then the the gene will not be transcribed. On the other hand, if the gene wants the products of that gene, the gene will be turned on, and then that gene will be transcribed. There are some genes which are constitutive genes, such as a constitutive enzyme, and this gene is turned on all the time, and it's expressed at a fixed rate, meaning the gene is transcribed at a fixed rate, so many per day, who knows. And that we call a constitutive gene. Other genes are expressed only when they are needed. And these are repressible and inducible genes, such as a repressible and inducible enzyme, where when the gene is not needed, it'll be turned off, and when the gene is needed, it will be turned on. And we'll talk a little bit about what repressible genes are and inducible genes, and then we'll talk a lot about one inducible gene, and that's the lac gene. We'll just make it the lactase gene. So regulation of gene expression can be pre-transcriptional control, meaning you can turn on or off the gene before the gene is transcribed. And that's one way to control the expression of that gene. We're going to talk about the lac operon, which is an inducible gene or an inducible operon. The lac operon is normally turned off, but when the gene is needed, it can be turned on. And that's what we mean by an inducible operon or an inducible gene. On the other hand, you have some genes which are normally turned on, and when they're not needed, they are repressed and then turned off. We're not going to be talking about repressible operons, and you will not be tested on a repressible operon. It's sort of the reverse of an inducible operon, and that is the gene is normally turned on, but when it's not needed, it can be turned off. There is also an epigenetic control mechanism where the gene can be turned on or turned off by epigenetic means. And this is really hairy to talk about because even today, microbiologists don't have a good understanding of why the epigenetic means is turned on the gene and how it turns off the gene. But when the gene is in a certain environment, the gene can be turned off or turned on. And like I said, it's really hairy to talk about, and it's a good thing we're not going to be talking about it and you won't be tested on it. Any question about any of that? And then there is another way that the bacteria cell can control its gene expression, and that is a post-transcriptional control. Like, for example, the gene can be transcribed, but before the messenger RNA is read into protein, the messenger RNA can be either inactivated or degraded. And that is a post-transcriptional control. We're not going to talk about that, and you will not be tested on that. So we're only going to talk about an inducible operon, and you will be tested on the lac operon, which is an inducible operon. Any question about that? And here's a question for you. If the cell doesn't need a gene product, does it make any sense for the cell to turn on that gene?
No.
Yes or no? Yeah, there's no reason to have the gene on if the cell doesn't need the gene. if the gene were to be turned on, the cell is just expending energy, making a gene product that the cell doesn't need. And so the cell will turn on and off genes depending on what the cell needs. All right. So when we're talking about gene expression, we're going to be talking about the operon model of gene expression. And a bacterial operon can be thought of as a super gene. In the super gene shown here, let me blow this up. We have the gene of interest. So this is actually the lac operon. And it does include the lactase gene, meaning the lactase enzyme, which is actually the Z gene here. That is called a structural gene, and that is the gene, the specific gene of interest. So Z is a structural gene. It codes for the enzyme lactase, the enzyme that digests the sugar lactose. Any question about that? Normally, when we are talking about a gene, like the blue-eyed gene or the brown-eyed gene, we're talking about a structural gene. An operon is a super gene that does include one or more structural genes, and it includes regions around those structural genes that control the expression of the structural gene. Specifically, two controlling regions we're going to talk about are the promoter and the operator, regions that control the expression of the structural gene. There's another region which is really short, and it's the terminator, and it ends the gene. So the terminator is the sequence of nucleotides at the end of the gene that codes for the RNA polymerase to fall off the DNA, ending RNA transcription. And then the promoter we've already talked about, so let me just repeat. The promoter is the region of the DNA upstream of the structural gene that codes for the RNA polymerase to bind to the DNA. And then the RNA polymerase moves down from the promoter and will be transcribing the RNA, transcribing the structural gene. Any question about any of that? Now, in the case of the lac operon, there are 3 structural genes in that one operon. In bacteria, there can be more than one structural gene in the operon. In eukaryotes, in an operon, there is only one structural gene in the operon. If it is a bacteria and there's more than one structural gene in the operon, all of these structural genes are related. So in the case of the lac operon, all of these structural genes are related to the metabolism of the sugar lactose. The Z gene codes for the enzyme lactase that breaks the sugar lactose into beta galactose and glucose. The Y gene is the gene that codes for the transporter protein in the cell membrane that will bring in the sugar lactose into the cell, meaning the sugar lactose does not readily get through the cell membrane. And to get it into the cell, you need a transport protein to pump it into the cell. And then the A gene is involved in the metabolism of the sugar beta galactose to the sugar glucose. all sugars are usually converted from whatever the sugar is to glucose, because glucose is the base sugar. And A gene helps the conversion of the sugar galactose to glucose. So all of the structural genes are involved in the metabolism of the sugar lactose. Any question about that? All right, so in eukaryotes, we obviously have an operon that codes for the enzyme lactase. All of those of you who drink milk products or eat milk products like yogurt, you have the enzyme lactase. So we have the structural gene A, and it will be in its own operon. I'm going to ask you a question. Do you guys have the protein that brings the sugar lactose across the cell membrane? That's a yes or no.
No.
You don't have a protein that'll bring the sugar lactose into your cell. So when you eat the sugar lactose, you're not going to digest it because it won't get into your cell. I'm
So then
giving you a
yes.
chance to change your answer. Yeah, we have the structural genes Y and A. It's just that instead of having them in one operon, eukaryotes have 3 operons for the metabolism of the sugar lactose. Any question about any of that? So eukaryotes only have 1 structural gene in their operon, but that doesn't mean that they have fewer structural genes. It's just that we have more operons. I guess that's an easier way to say it. Because for the lac operon, we don't have 1, we've got 3 operons to metabolize the sugar lactose. Any question about any of that? All right. This graph is showing you a regulatory gene, I. This is not part of the lac operon. It in fact is its own operon, which is not shown here, but right there would be the regulatory gene operon. It'll have its own promoter and own operator, its own terminator. And so it has its own operon. The regulatory gene, what it does is it makes a protein which binds to the operator right there in the lac operon. And when that protein binds to the operator, the RNA polymerase, which binds to the promoter of the lac operon, cannot then move down the DNA past the operator because it's blocked by the protein. And then that shuts down the lac operon, meaning the lac operon will be turned off. And how that's done is by the regulatory gene making a protein that binds to the operator. And that's telling you what the function of the operator is. It's also telling you the function of the regulatory gene I. Normally, in both bacteria and eukaryotes, the regulatory gene is nowhere near the gene that it regulates. In fact, in eukaryotes, usually the regulatory gene is on a different chromosome than the gene that it regulates. But in E. coli and in bacteria, with the lac operon, the regulatory gene is actually right next door to the gene that it regulates, meaning the lac operon is right next to the regulatory gene. Any question about that? Oh, here's something else I need to talk about. Eukaryote operons are similar to bacterial operons, except they only have 1 structural gene in the operon, but they have more regulation of the operon than a bacterial operon has. So, for example, there could be 2 regulatory proteins. There could be more than 1 regulatory controlling region. We won't talk further about that. All right, the regulation of gene expression can be either induction, which the lac operon is a case of, where the gene is normally turned off, or it could be repression, where the gene is normally turned on. In repression, the gene can be turned off when the gene isn't needed, and in induction, the gene can be turned on when the gene is needed. The lac operon is an inducible operon, so it follows induction. Let's go back to the previous slide. E. coli, which is normally found in the soil or on an apple, will not be around the sugar lactose. So E. coli oftentimes will not be around the sugar lactose. If E. coli is not around the sugar lactose, why would it want to turn on the lac operon? It doesn't want or need the lactase gene or any of the other genes involved in the lac operon because the sugar lactose is not around. And that is why, at least in E. coli, the lac operon is normally turned off. But let's say that you are an E. coli on an apple and somebody picks the apple and then consumes the apple, getting the E. coli into a gut. And we're just going to say a teenager is the one who ate the apple. And in this teenage gut, the teenager drinks a lot of milk. The E. coli is now going to be around the sugar lactose because this teenager is drinking a lot of milk. Any questions about that? So the E. coli is going to want to turn on the lac operon because the teenager has a lot of milk and it's his gut or her gut. And the lac operon is inducible, so it can be turned on. How it's turned on is the repressor protein binds to the operator, shutting down the gene. But in the case where there's the sugar lactose around, there is allolactose around. Allolactose is an isomer of lactose. So one is the right-hand image and the other is the left-hand image. I think lactose is the left-hand image, but I could be wrong. Don't quote me on that. And allolactose is the right-hand image. The point is, whenever you have the sugar or lactose around, you have allolactose around. Allolactose is the inducer. Allolactose can bind to the repressor protein and inactivate it. If the repressor protein is not activated, it cannot bind to the operator. And then if the operator is free, the RNA polymerase can bind to the promoter and then transcribe the Z, the Y, and the A genes. Any question about any of that? All right. So how is the repressor protein removed from the operator? And I'll give you a hint. It is not capable of binding to allolactose when the repressor protein is bound to the operator. So allolactose does not bind to the repressor protein and inactivate it when the repressor protein is bound to the operator. Anyone have a guess? All right, the repressor protein is binding to the operator by a stoichiomic reaction, and every once in a while, the repressor protein falls off. If there is repressor protein around, it will bind to the operator, shutting down the lac operon. And then this polymerase right there, when it binds to the promoter, is it going to be staying here until that repressor protein falls off? Meaning, is that polymerase going to be stuck there literally, potentially forever? No, that polymerase also binds to the DNA by a stoichiomic reaction, and every once in a while it can fall off. And once it falls off, it can then bind to another promoter and then begin protein transcription of that gene. Of course, if it binds to the lac promoter again, it will once again be blocked by the repressor protein. But if the repressor protein falls off and allolactose is around, allolactose will inactivate the repressor protein that's fallen off, and then there's no active repressor protein to bind to the operator, and that will turn on the lac operon. And then the lactase gene will be transcribed. Let's watch a little video on the lac operon. If I can get that to work. Boy, that's 3 times I had to click on that.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon: beta-galactosidase and permease. Gene expression can be induced or turned on.
Let me talk about these 2 proteins.
Beta-galactosidase and permease.
Beta-galactosidase is the same thing as lactase, just another name. And then permease is the transport protein that brings the sugar lactose across the cell membrane.
Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region.
That's the operator.
Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region.
All right, that's a little bit of an oversimplification. The lactose does not bind to the repressor protein when the
controlling region
is.
At this point, RNA.
Come back here. The repressor protein does not bind to allolactose, which isn't mentioned in this video, when the repressor protein is bound to the DNA, the operator. Like I said, this is a little oversimplification. It binds to the repressor protein when the repressor protein is not on the DNA, inactivating the repressor protein. And then the repressor protein cannot bind to the DNA, the operator.
Lactose is the lac-operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac-operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac-operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions about that video? All right, when we look at this messenger RNA, what's unusual about it? Have we ever seen a messenger RNA like this?
No.
Yeah, it's coding for 3 different proteins. So we've never talked about a messenger RNA like that. But in the case of the lac-operon, that messenger RNA actually codes for 3 different proteins. Beta-galactosidase, also called lactase, permease, and that's the transport protein that brings the sugar lactose into the cell, and then transacetylase, which I'm not going to talk about. It helps convert beta-galactose into the sugar glucose. All right, any questions about the lac-operon or operons in general? All right, not hearing anything. So let's change gears and we're going to talk a little bit about mutation. A mutation is a change in the genetic material. A mutation may be neutral, it may be beneficial, or it may be harmful to the individual or cell in which the mutation occurs. Many, in fact, most mutations are harmful, although many mutations, strangely enough, are neutral. We'll talk why in a minute. And there are only a few mutations which are actually beneficial. Here is actually a mutation shown. And in a sense, this is a mutation that happened 10,000 years ago in Northern Europe. Before 10,000 years ago, the only human eye color that humans had were brown eyes. And 10,000 years ago in Northern Europe, we had a mutation, which was the blue-eyed allele was created. And now the blue-eyed allele actually in Northern Europe is the predominant allele, more common in Northern Europe than in the brown-eyed allele. The blue-eyed allele is rare in populations outside of Europe. Not Europe, not Europe. I'm trying to say Caucasians. But it does happen every once in a while in other populations. And that happens probably, well, I'm not sure when it happened. It might have been a mutation that made the blue-eyed allele. I must have my mail program on, so let me shut that down. I just got a email. And that is, I have to go there to open up the Canvas. Otherwise, I can't open up Canvas, and that's where I have my link for starting Zoom. All right, where were we? So mutations can be neutral, beneficial, or harmful. And the blue-eyed allele is an example of a mutation. And there's another example of mutation where this individual has partially brown eyes and partially green eyes. A mutagen is an agent that causes mutations. However, a mutation can happen without a mutagen, and that we call a spontaneous mutation, a mutation that occurs in the absence of a mutagen. A spontaneous mutation can happen simply when the DNA polymerase is replicating the DNA, and it inserts the wrong nucleotide. That can lead to a spontaneous mutation. There are different types of mutations. There's a base substitution, also called the point mutation. And these can be a silent mutation, a missense mutation, or a nonsense mutation. I'll talk more about these in a minute. The difference is only one nucleotide base is changed in a base substitution. There's also a frame shift mutation, and this is when a nucleotide is added or a nucleotide is removed from the DNA, and it's called a frame shift mutation because when you remove a nucleotide, you change the codons after you remove that nucleotide. Not only do you change that one codon, but you change all the codons after that codon. And so it results in a frame shift mutation that the protein will be totally different from that frame shift mutation. And then there's a macromutation, and this is where an entire region of a chromosome is added or deleted. A macromutation is not discussed in your book, so pay attention to the lecture when we're discussing a macromutation. And I will warn you that the internet talks about two different types of macromutations. One macromutation is the one I'm talking about, where an entire region of a chromosome is either added or deleted. And then the other macromutation is where a controlling gene is mutated, and so it causes many different mutations to happen, meaning the controlling gene controls other genes, and then when the mutation happens, many different characteristics of the organism can change. And that macromutation I'm not going to talk about because that's a controlling issue. I'm talking about where a very large chunk of DNA is either added or deleted. Does anyone know a macromutation where an entire chromosome is extra?
Down syndrome?
Yeah, Down syndrome. Where the individual has, is it chromosome 21 or chromosome 23? I seem to forget that. I think it's chromosome 21. The individual has 3 chromosome 21s. Is that right? Can anyone confirm that?
I think it's 21 too.
Okay, great. I know it's either 21 or 23. I don't remember which. I think it's 21 though. Another macromutation is an individual which is born with only one X chromosome, or an individual born with three X chromosomes. The three X chromosomes individual will be phenotypically normal. The one X chromosome individual will be not phenotypically normal. They'll be similar to the Down syndrome patient. And then another macromutation is an individual born XXY. This individual is actually what the Greeks call the hermaphrodite, an individual that can be born with both male and female body parts. Usually one part dominates over the other, but in a pure hermaphrodite, the individual can be half female, half male, and usually the body is divided in half. And then another macromutation is an individual born XYY. These individuals are phenotypically male, and most of the time they are phenotypically normal, meaning you have to do a chromosome comparison to know that they are XYY. So there are some macromutations that do happen in the human population. And some of them, like the XXX individual and the XXY individual and the, well, maybe not the XXY individual, because hermaphrodites might not phenotypically be normal, but they are capable of reproducing. And the XYY individuals are phenotypically normal too, and capable of reproducing. So some macromutations happen usually when an entire chromosome is removed or added. That will be lethal to the embryo of the individual. Like if the embryo only has one chromosome 1 or 3 chromosome 1s, that will be lethal to the embryo. But there are a few individuals which can have a macromutation, and then they're even phenotypically normal. And the best example is XXY, and that's because XX is female, XY is male, and so an XXY individual is phenotypically normal, except that they have both male and female body types. By phenotypically normal, I'm talking about capable of reproducing, and phenotypically mentally normal. Any question about any of that? All right, let's talk a little bit about base substitutions, where the DNA, when it is being replicated, a mistake can be made where the DNA polymerase adds the wrong nucleotide. When this DNA now replicates, that will be, instead of a GC pair, that will change to an AT pair, and this mutation will then become fixed. If this doesn't actually hydrogen bond, so the cell could recognize this as an error and then remove the newly added nucleotide and put in the correct nucleotide. So before this gets replicated, the cell could correct that error. And what the cell does, it simply assumes that the newly replicated base is the problem, which most of the times it is, and then it can just remove that nucleotide and then insert the correct nucleotide. So the point is the cell can correct this as long as it corrects it before the DNA is replicated. But once the DNA is replicated, it fixes this mutation. And this mutation here changes the amino acid. So obviously that'll mutate the protein. Any question about any of that? All right. So that was a base substitution where one nucleotide gets changed, and this is actually a missense mutation. We'll talk a little bit about that later. Let's talk about a silent mutation. A silent mutation, also called a neutral mutation, is where the DNA is changed by one nucleotide, but it does not result in the protein being changed. And the reason is because the codon is changed, but the codon codes for the same amino acid. Like if UUU is mutated to UUC, that still codes for the same amino acid. And this is called a silent or a neutral mutation, where the protein is not changed. The DNA is changed, but the protein is not. So there's no effect on protein function. And that's why it's called a neutral mutation or a silent mutation. Any question about any of that? Let me go back to something, and that is 10,000 years ago in the human population, we had the mutation in Northern Europe of the brown-eyed allele being mutated to the blue-eyed allele. Today, when we look at those two different alleles, they are neutral, meaning the blue-eyed allele has no reproductive advantage over the brown-eyed allele. Now, in the past, the blue-eyed allele may have had a reproductive advantage. And that's because in Northern Europe, the blue-eyed allele became the predominant allele in Northern Europe. And that means it either had a reproductive advantage or it was linked to a gene which gave the blue-eyed allele a reproductive advantage, meaning the blue-eyed allele happened to be next to a gene that gave the individual a reproductive advantage. And that way the blue-eyed allele got carried to becoming the main gene in Northern Europe. But today, when we look at it, the blue-eyed allele and the brown-eyed allele are neutral. Neither one has a reproductive advantage over the other. And that can be another neutral mutation, where simply there's two phenotypes, but both phenotypes have the same reproductive advantage. So it's a neutral mutation. Any question about neutral mutations? All right, a missense mutation is where one changes the DNA base and it results in a change in an amino acid in the protein. So here we have the codon GGC, and it can be mutated to the codon AGC simply by changing one nucleotide right here, changing from G to A. Instead of coding for the amino acid glycine, we're coding for the amino acid serine, and that would be a missense mutation, where the nucleotide is changed in the DNA and the amino acid is changed in the protein. And that is a missense mutation. Some examples of a missense mutation is sickle cell anemia, which can be a missense mutation. Another missense mutation was the blue-eyed allele. All you have is one nucleotide change from the brown-eyed allele, and it resulted in a different protein, different color of the protein. we won't talk about how the color comes about, but change the eye color from brownish to bluish. Any question about any of that? I guess the next question is, can humans have another mutation in their eye color to get yellow eyes? If you do not know, dogs have brownish eyes and bluish eyes and yellowish eyes. So it's possible sometime in the future, humans may have yellowish eyes. We need a specific mutation though. All right. A nonsense mutation is where you change one nucleotide base in the DNA and it doesn't change the amino acid. It codes for a stop codon, and that will make a truncated protein. Let me blow this up. So here we have the codon AAG coding for the amino acid lysine, and then we have a very small protein only having 4 amino acids, and we change the A here to a U, coding for a stop codon, and now we have an even smaller truncated protein only having 1 amino acid, which is not very likely, but it's very unlikely going to have a protein with only 4 amino acids. So what can we say? The point is, it's a nonsense mutation where the nucleotide is changed, resulting in a stop codon, which causes a truncated protein, a smaller protein. Any question about any of that? There are some muscular dystrophy patients and cystic fibrosis patients that have a nonsense mutation where the nucleotide is changed, resulting in a stop codon, and then these individuals make a truncated protein, causing the disease, muscular dystrophy, or cystic fibrosis. Any question about any of that? All right, let's move on to talking a little bit more about frame shift mutation. A frame shift mutation is where you insert or delete one or more nucleotides into the DNA. When you have a frame shift, like right here, we had an A pulled out, it changes not only this amino acid, but all amino acids after that. And the reason being is that the codon is AUG, AAG, and now it can't be UU, what was that before? UUU, and it just borrows the next nucleotide. And so the U has been pulled off, and so it now becomes UUG, and then the codon isn't going to be GGC anymore. It's going to be GCU, meaning it just reads the codons in groups of 3s. And so whenever you have a frame shift of 1 being added or 1 being removed, you change the DNA from the point of that frame shift, not only changing this amino acid here, but all of the amino acids after that point. And that's what a frame shift mutation is. This can happen when you remove or add 2 nucleotides. You'd have AA being pulled out. Come on here. Come on, mouse. So instead of having UUG, you then have UGG. And that would change the amino acid there too. And then everywhere else. However, when you have 3 amino acids either added or removed, you only change 1 amino acid, meaning you remove UUG, come on, mouse, you'll have no phenylalanine, but you'll have the glycine and then the stop codon. So it does change this one amino acid, but none of the other amino acids afterwards are changed. So a codon of 3 or a multiple of 3, either being added or deleted, is not a frame shift mutation. A frame shift mutation is removing 1 or 2 or 4 or 5 or some multiple of nucleotides other than of some multiple of 3. And that's a frame shift mutation. Any questions about frame shift mutation? Now, a frame shift mutation really changes the protein, because after the frame shift, all of the nucleotides can be different. All right, any questions about any of that? How are we doing time-wise? Oh, oh, here it is here. Let's just go on a few more slides. All right, so there are two causes of mutations. There can be a spontaneous mutation where the DNA polymerase simply makes a mistake and inserts the wrong nucleotide base. There's no mutagen causing the spontaneous mutation. And then there can be a mutagen where the mutagen causes the mutation. There are two types of mutagens. There are radiation and chemical mutagens. There are two types of radiation that can cause mutations. There's ionizing radiation, and this is radiation with a high energy, like X-rays or gamma rays, and when they strike an atom or a molecule, they give the atom or molecule enough energy that the electron is actually removed from the atom or molecule, and that will create an ion. So this will now have a positive charge, and that's an ion. This ion can then react with DNA, causing the DNA to be mutated. And that's how ionizing radiation changes the DNA. It just strikes either a molecule or the DNA, causing the electron to be left from the molecule or the ion. And then this is an ion, which can then bump into DNA, mutating the DNA. There's also non-ionizing radiation, and there's more than one form of non-ionizing radiation. Visible light is a form of non-ionizing radiation. It doesn't cause mutations, but UV radiation can cause mutation. UV radiation causes thymine dimers, where two thymines, which are next to each other in the DNA, can cause a dimer, two covalent bonds form between the two thymines, and that's unusual. Usually the thymines are not bonded to each other. The two thymines that have the dimer now cannot hydrogen bond to the two adenines in the complementary strand. That will cause a bubble in the DNA as shown here. And the DNA polymerase cannot replicate this DNA, and or at least not replicate it correctly, and the RNA polymerase cannot transcribe this gene. So this thymine dimer really screws up the DNA. Any question about any of that? Anyways, UV light can cause a mutation in the DNA. When the UV light strikes the DNA, it causes two thymines to form two covalent bonds between them, causing the thymine dimer. Any question about any of that? There's also a chemical mutagen, and we're only going to talk about one example, and that is nitrous acid as a chemical mutagen. Here we have our normal DNA. When nitrous acid binds to the DNA, it can convert this adenine. It still is an adenine, but it will change the way this converted adenine will bind to. This converted adenine does not bind to a T, and the DNA polymerase, when it's replicating that modified A, will not read it as an A, it'll read it as a G, and then insert the C next to that modified A. So it will, in a sense, mutate that A there and make it act like a G. And then when this gets replicated, that will fix the mutation. Instead of having the AT nucleotide pair, we'll have a GC nucleotide pair. And that's how nitrous acid acts as a mutagen. It actually binds to this A, modifying it so this A does not act like an A, it acts like a G, mutating the DNA. Any questions about any of that? I sort of lost my place here. Oh, there it is. All right, if there's no questions, I'm going to end the slide here, and I'll see you in the lab at 7. Bye.